The next guest will introduce us to an innovative technology that can be used to mark and detect materials and products. In addition, he will introduce us to the sorting technology, tracer-based sorting, TBS. Peter, why are materials and products fingerprinted? It is so easy to counterfeit or copy a product. What do you think?
Okay, the anti-product pirate issue was the first topic of this tracer project. But now comes another aspect of these markers. With a few milligrams per kilogram of these additives compounded in the material, you can identify the polymer for what it is used for, food or automotive, and even from which company it comes. That opens up a completely new view on quality of recycling and a tremendous possibility.
There's a lot of potential, I guess.
Absolutely. Yeah.
Thank you, Peter. And now I would like to welcome Jochen Moesslein from Polysecure GmbH with a presentation on this topic, new sorting and tracking options based on innovative marker technologies. Enjoy the film.
Hello, everybody. I'm very happy to be with you today and present our technology options for biopolymer-based products. On this very first picture, you see already a bit what our vision is regarding sorting technology. The plastic items and also other material items and materials could be clustered into specifications, into specific fractions, and then marked by specific sorting tracers, fluorescent sorting tracers, and the sorting could then be done according to the sorting tracer and would provide precise sorting, reliable sorting to whatever the definition of the sorting fraction is. You see in the picture that I have included a fraction for PBS biopolymer. This, of course, can be generalized to other materials, and if that would be done like this, then we could sort the PBS biopolymer items and materials to its specific fraction. You will see more on that later. One slide on our company, Polysecure. Polysecure is a technology company, a little bit later than a startup, 10 years old. We are 40 people with a pretty strong scientific engineering base, chemists, physicists, engineers, but also business development managers. We have one approach, or one-stop approach, meaning that we develop on the one side the marker materials themselves, but also the devices to detect the marker materials or sort materials. As typical for a technology company, we have some patents. By now, it's about 30 patent families, and we have strategic cooperations with scientific research institutes such as KIT or Institut Charles Hadron. But we also have a very strategic cooperation with Carl Zeiss regarding especially the sorting technology. We have an advisory board with people from industry, from politics, and from science. And we do a lot of research projects with partners, with industrial companies. You see a few names in this section. What are our applications? Our applications is 4 applications today, from left to right. On the left side, this is our sorting technology. We labeled it tracer-based sorting because quite a bit of the sorting could be done based on tracers, but in that machine, which is equipped with a so-called combined detector, we not only measure the tracers, we also do NER reflection spectrometry. We measure the color. We take a picture with a Wiz camera and process it with artificial intelligence. And we could optionally also measure watermarks or other sorting features. So this is the concept. We call it TBS Complete. where the sorting of materials and items is done at one spot and within this one spot or process, the item or the material is completely characterized and then transported to its proper final destination with a system that is very comparable to letter sorting. The second application, right of this picture, you see a hand with what we call excitation device. And what you see here is a picture of our fluorescent particles, our up conversion fluorescent particles in a material matrix. Could be a biopolymer. The excitation is done in the near infrared, and the material, our particles, react with visible fluorescence. And as some of you may know, and it's pretty obvious that this is a bit against energy conservation. So no other material can do such a energy transition, taking an excitation wavelength, a longer wavelength, and then convert it to light in a shorter wavelength is normally not doable. It's only doable with the two-photon process. And therefore, the contrast of our fluorescence within materials is very good. And we exploit that and say, well, this generates a fingerprint, and we can register this fingerprint, and this fingerprint is unique for every item. So we have a unique identifier for every item. The third application, right of this picture, is PolTech. PolTech is a technology to track materials. It is based on macromolecules, and for the first time, a very efficient and very controlled synthetic process to produce macromolecules with an exact sequence of monomers. And we use those and blend it into materials and identify and measure these materials then by the mass of the monomers of the macromolecules. You will see later a bit more on that. And to the very right is our fourth application. It's actually the same fluorescent particles as the two applications before. We call it brand-proof. It's a device by which you can very quickly, very small device, you can measure the fluorescence of our fluorescent tracers and use it for originality detection and anti-plagiarism. A slide on our marker materials. You may ask, what are these materials? There is of course a broad portfolio of materials, but we choose materials with very good thermal stability, especially for the fluorescent materials. Most of these materials today are inorganic and are very temperature stable. They are annealed or sintered above 1000 degrees Celsius. We can tune the particle size, and the chemical inertness is also very high. So we have good biocompatibility for these materials, and this results in very good tests, toxicological tests. And of course, the struggle is always to have precise detection with low concentration of marker materials. And this is given by research, by optimization, and of course also by optimizing the quantum efficiency, especially for the fluorescent tracers. And you see a line below, especially with the fluorescent materials, they were already dispersed in end materials with a volume of 70,000 tons, maybe by now almost 100,000 tons. So these marker materials are well tested, one could say. Now I'm turning towards tracer-based sorting. You see here a closer picture of our sorting process. And again, you see this combined detector on the one side. And this is really the strategic collaboration between Polysecure and Zeiss, a new device. How does it actually work, tracer-based sorting? So you have the fluorescent tracer materials, you integrate it in the bulk of a packaging material or packaging item, or you can integrate it via ink onto the packaging. You see here a picture, and you even see some fluorescence on that label. And then you have the fluorescence integrated basically in your material, in your product. You move the product or the material through a laser curtain with conveyor belts. The laser curtain or the laser light will excite the tracers to fluoresce, and the fluorescence is measured with cameras, and the signals are processed and correlated to a specific sorting fraction. There can be quite a few sorting fractions. You may ask, how many variations can you create with these kinds of fluorescent tracers? The number today is 100. So we feel confident to produce or generate 100 different sorting codes and reliably differentiate these sorting codes. You here see a video within our sorting device. Maybe you can see the speed of the packaging devices, how they are moved through the detection area very fast, and nevertheless, the measurement is very precise because, of course, fluorescence is an isotropic emission of a signal. It's not like picture recognition, where you have a picture on a structure or on the surface of a device, and you have to recognize the picture. Then you have to direct the product or the picture towards the camera. Here is totally isotropic, the signal, and that makes it easier to work with deformation, with high speed, and other typical challenges for packaging products. You now see a film of our demonstration device. It's a demonstration device that shows what happens in a sorting machine. We have packaging devices with 100 microgram per packaging device on the surface via ink. And in the system, the packaging is moved through the laser curtain, which is above, on top of it. You here see in the dark the fluorescent light. So the packaging is moving through the laser curtain and, of course, emits fluorescent light. You can here see that it's only a very small area that is equipped with the fluorescence, and the fluorescent tracers are embedded in the ink here. And of course, you can move the packaging at a higher speed, at 3 meters per second, through the device. You see it moves from left to right, from right to left, and according to the tracer code, a picture is drawn up indicating what type of packaging material has been sorted. Here you see an animation of the sorting process. So very important in our sorting process, we first singulate the items, then we shuffle it through the combined detector. So it's only one sorting stage, not multiple stages as the state-of-the-art technology today. And then every item is directed towards its final destination. And already at this picture, you can see that, for instance, if the market decides to put up a fraction for specific biopolymers, then of course this can be a final destination. The incremental extra cost of sorting another fraction is very small. It's very small. So this system is really flexible for new materials, for new sorting fractions, so that they can also be sorted. So advantages at a glance. The sorting technology is the first technology open sorting technology because it measures in a combined detector, NER, color, tracer, watermark, and takes a picture. So whatever sorting feature is on an item, we will measure it and we can use it for the sorting decision. The items and the materials are not blown out by pneumatic valves, but it's a mechanical system which is much more precise. So there's a very high purity of the sorting fractions, very little mixtures, very little, almost no false positives. So this is very important if you want a pure end stream. So it's really a purification. And the single line process is very flexible to new fractions. I think that became clear with the picture before. Now I'm moving to the second application, the particle fingerprint application. The same particles actually as the particles that are used for the sorting execute this star-like picture in every material if you want. How can that be used? First of all, if the particles are embedded in the plastic material, you have to register the product. Every product has to be registered for its individual fingerprint. Then it's registered, and when it comes to another detection station, or maybe at the end of its life, then it's detected and measured again, and then correlated to the registered fingerprint. So you can think of a circle. You can think of the fingerprint being, for instance, in a bottle, the bottle being registered, the bottle being sold to end consumers. The end consumers bring it back to a sorting facility or a deposit system or a deposit machine. In the deposit machine, the particle fingerprint is measured, and maybe accordingly to its life, maybe the bottle has been filled up with an ecological orange juice or whatever. So according to its specific life, there can be an incentive or something like a deposit paid back to the consumer. So these fingerprints enable individual incentivization schemes. When the bottle is then recycled and grinded and compounded, the fluorescent particles will remain in the compound. And when a new bottle is blown out of this marked compound, then of course you have a new fingerprint. So basically the fluorescent material remains in the compound. In that regard, the compound is sortable by its color, by its emission spectrum, but the particle fingerprint is generated new from every new extrusion and blowing process. Of course, we have almost an infinite number of particle fingerprints, and you may have understood that it's almost impossible. It is impossible to copy such a fingerprint. How could you do that? The particles are embedded in the material, so it's not comparable with something that's printed on. Something that is printed on, like a barcode, you can dissolve with a washing agent or water or a little bit of a solution. You can wash it off. This is not possible with the particles. They are really included in the material. And this is a big advantage. As you may remember from the introduction, Poltec is our material authentication technology. It goes back to macromolecules. So a little bit the same as with the fingerprint. The Poltec actually is a solid material. It's also a whitish powder, a solid material. It can be blended into the compounds at very small concentrations. The magic number is 1 ppm, so very little. You blend it and homogenize it into a compound, can also be a liquid. Then the product is produced out of that compound, and at various stages of a potential circle, you can measure the Poltec mass code. You would have to take a little bit of material, very little, in the microgram area, and then you would do a tandem mass spectroscopy, and by that mass spectroscopy you can identify the material. So you can backtrack it to its production batch. You can of course look how pure a material is or how much recyclate content is included in the compound. Yes, I think I've mentioned already quite a few of these advantages, and the number of codes that are possible with Poltec is also almost infinitive. The codes are generated with a combinatoric mechanism. and it's extremely difficult to re-engineer these macromolecules because the variation possibly are so huge. So it's also very safe. Our latest application in this presentation is the brand proof. This is the device we have developed. So we have developed the hardware and the software and everything of this device. You here see the detector laying on a decorative foil. This brown foil at the below picture is a foil. And there are millions of square meters of these marked foils out there in the markets. And with our little device, basically on every square centimeter, there are few particles, and these few particles are sufficient to identify the particles with this precise measurement of the decay constant. This is a measurement of the decay constant of this specific fluorescence. The advantage is, of course, very mobile measurement, very quick measurement, and interesting, the particles are also the same as for the tracer-based sorting. So now let's look at circular economy applications of our technology. We here depict a typical cycle. So at the left above side at approximately 11 o'clock, you have the feed-in of raw materials, renewable raw materials, fossil raw materials. So thinking of biopolymers, it would be a renewable feedstock. You use it to compound the final material. The material is used for the manufacturing of products. Could be packaging devices, but could also be other products made out of the compound. And the compound is brought into use. And in the middle of the circle, you see this area with Polysecure and the two types of marker system. One marker system is the fluorescent tracer, and the other is the Poltec macromolecules. You can include these at the compounding stage and the material processing stage, or even at the production state. You can include both marker materials via a master batch, for instance, into the production of final materials. Now, during use and collection, of course, you could measure the product or detect the product based on these marker materials. But very importantly, at the sorting stage, you can exploit especially the fluorescent tracers and sort the material and products, and then direct them towards the best possible recycling pathway, be it mechanical recycling, be it solvent-based recycling, or even chemical recycling. So we see our sorting technology as a little bit of flag system, a sorting system that interconnects the recycling side of the circular economy with the production and use side. And here we are at my last slide, what Polysucker can do for biopolymers, a little summary. We've talked about tracer-based sorting. Tracer-based sorting would enable closed loops also for the different biopolymers. A typical tracer concentration is 10 ppm, and we have something like 100 codes per polymer, so it's quite a bit. Then we have talked about the particle fingerprint technology would enable individual tracking of biopolymer products for product pass, something that is pushed by the European Union, for instance. Product passes will come. And interestingly, TBS and PFP can be based on the same particles. Then Poltec is for material tracking, batches, formulations, purity, recycled content. The required concentration is lower, 1 ppm. And then we have talked about brand proof. So I'm looking ahead to the discussion, and I hope I could convey to you a bit about our technology platform, and happy to discuss further with you. Thank you very much.
Thank you very much for your interesting presentation, Mr. Moesslein. Unfortunately, he can't be with us right now. Instead, we are happy to welcome Reiner Just to our live discussion. Hello, Reiner Just, and a very warm welcome from our side. from
Yes,
Merseburg.
welcome.
How are you?
Hello, welcome. Yeah, nice to see you. Well, I'm fine, thank you.
Again, here we got lots of questions. And I take the first one because it's something that I'm also interested. Do you provide master batches with these tracers? If yes, what are the matrix polymers?
Oh, we can produce the master batches, that's correct. And we can produce in almost every polymers we get. If there are special polymers, we sometimes ask our customers to send us the basic polymer to batch them.
Okay, so and the second question is, are there any limitations for the sorting of biopolymers in comparison to conventional plastics?
No, not as much as I know. As we've seen in the presentation, our tracers are very stable for heat and mechanical influences, as well as chemical influences. So I don't see any difficulties to use it in biopolymers.
Okay.
Great.
So now comes a tricky question.
Okay.
Okay. What about what about food legislation and certifications for composting?
Yeah. That's not a tricky question. It's quite an easy question.
Okay.
We are still in the process to get the food contact for EFSA, and it's still in progress. But as Mr. Moesslein said, we did all the toxicological tests, and they were very positive. So we haven't had any problems. That's why we believe to get all the food contacts. But it takes time.
So it seems to be that you are optimistic.
Yes, we are optimistic, of course. Because the first sign from the toxicological tests are very, very good. But as it always is, it takes time to get all stuff done and to get the papers. So normally it takes 2 to 4 years to get there's a certificate.
I see. Good. So let's see what the next question is here. What about recycling of the materials with the tracers? Are there any effects of recycling?
No, there aren't any effects. The positive thing of these tracers are that they are still in the material during the recycling process. So once we have decided which material gets which coat, this coat will always last in the material, and it will last for many recycling turns.
Okay.
That sounds good, definitely.
I think so. The first thing which needs to be done is to standardize the materials for the special tracers.
Okay.
You need to get a standardization.
I see. So thank you very much for answering the questions and for being with us today instead of Jochen Moesslein. And yes, we're wishing you all the best. And thank you for being with us.
Yeah. Thanks a lot for your contribution to this Biopolymer Congress. I'm pretty sure that it will be a let's say, an important role of these tracers in the industry.
Yes.
I thank you. Thank you for being with you. And have a good time.
Thank you.
Thank you very much.
Bye-bye.
Bye.